So far, our look at enzymes has allowed us to understand their structure and the fact that they're complex protein molecules held together by hydrogen bonds, and the shape of them is extremely important. In this video, we're going to look at some of the factors that are in systems that can affect the ability of these enzymes to work. And the first one of these factors that affect enzyme function is temperature. Living systems have a certain temperature, like our body temperature is roughly 37 degrees Celsius, and the enzymes that are in our body function really well at 37 degrees Celsius. But if our body temperature changes, that can affect the ability of these enzymes to work. So let's have a look at that particular thing. I'm going to represent my enzyme over here, the same sort of thing as I've drawn before, just so I can refer to it. This enzyme is a complex protein. It has an active site somewhere on its surface, and it's held together by hydrogen bonds, some disulfide bonds as a type of covalent bonding, and there's some perhaps ionic bonding in it too. But that's the sort of thing we're talking about. Now what happens with temperature is if you increase the temperature in a system, what that does is it affects these molecules. The hydrogen bonds can break. Increasing temperature can break hydrogen bonds. And if that happens, some part of this molecule may be free to move around a little bit. It's going to change the shape of this molecule. That'll change the shape of the active site. And what we had already is that the shape of the active site is paramount in its function. If the active site changes shape, this thing won't work. So here's what we have with temperature. It can always be represented as a graph. If we have this as the temperature across here, put in degrees Celsius, because I'm going to use 37 as an example for our own body temperature. And at 37 degrees, the enzymes in our body are working quite well. So this is the rate of enzyme activity. So all those enzymes that are involved in our metabolism, all the enzymes that work in our cells to make the molecules we need, the enzymes that are in our food that are digesting the food, and that sort of thing, they work really well at 37 degrees. But like I say, if you increase the temperature, hydrogen bonds can break. That's going to affect the rate of the enzyme's function. And what we get is this sort of a pattern here. At increasing temperature, the ability of the enzyme to function decreases. If you continue to increase the temperature, it's going to hamper the function more and more and more, and pretty soon it's not going to work at all. It's going to be denatured. So it's going to drop right off. So it will cease to function. And in our bodies, many enzymes will actually cease to function 45 degrees, that sort of thing. If we had maybe 50 degrees, somewhere in there, the enzymes just simply aren't going to work. So this is where the enzyme is denatured. This is where the enzyme isn't working at its best. The best is called optimum. That's right here. This is its optimum. Now just to illustrate this, make sure that you actually understand this using a really simple example. Our body temperature is 37. I'm sure that every one of us has had a fever. When you have a fever, your body temperature is elevated. So you may be 37 degrees, 38, 39, 40 degrees. At 40 degrees, you're not feeling very good. Why aren't you feeling very well? Well, your enzymes aren't working very well. And if the temperature continues to go up, so if you go up to 40, 42, 43, sooner or later you're going to denature some enzymes. If you denature the enzymes, they lose their tertiary structure, or in some cases quaternary structure, and you've lost it. They cannot be recovered. This thing's going to change shape, and part of it will loop back on itself, maybe bond in a different fashion, and that molecule ceases to function. Eventually, your body will, if the temperature goes back down, your body will generate new enzymes. You always have the ability to make the enzymes, but you don't always make them because they're used over and over again. I only drew half the graph here. If you get into not a hyperthermic condition, so hyperthermia is when you're too hot, you get into a hypothermic condition where your body temperature is going down, 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 maybe 34 degrees might be considered hypothermia. The enzymes aren't working so well. The reason they're not working so well is because these things are becoming more and more solid, and the requirement for an enzymatic reaction was an induced fit, if you remember. This thing had to give a little bit of a shape change, a little flinch to get the reaction to work. Well, if it's too cold and it's too solid or too solidified, that won't work. And also, if things get cold, the system cools down, the molecules simply don't move around as much as they should. So you're losing kinetic energy. Loss of motion of molecules. They just don't enter the active site, they don't leave the active site, that sort of thing. So the rate of reaction is going to go down again. So even on this side here, where temperature is decreasing, the reaction rate decreases. But notice it's not a symmetrical graph. There's no denaturing on this side. This is going down due to a loss of kinetic energy and a loss of the ability of the enzyme to perhaps do that little induced fit bit. Right? So there's one thing that affects the rate of enzymatic activity, temperature. As temperature increases past optimum, the enzyme rate will decrease. At some point it's going to denature. It'll cease to function because it just cannot conduct the reaction anymore. It's too deformed, if you will accept that term. On the lower end, it has not denatured. It is possible on this end that as the temperature increases, it gains back the kinetic energy. Any deformation of the enzyme can be restored, so it can actually renature on this side. It can get back to normal from here. If there's a loss of enough kinetic energy and the temperature is down so far and hypothermia sets in and a system actually gets too cold for too long, then that's really detrimental. There are situations where you could look some examples up where people have been literally frozen, almost, they've been so cold and they've recovered. In other cases, people get that cold and they do not recover. So that's temperature, one of the factors that affects enzymatic activity. The second one that I'm going to deal with is pH. I'm going to get rid of that box here. Hang on. And I'm going to number them. So we've got temperature is the first factor that affects enzyme activity. Factors that affect, we're talking about the rate of enzyme activity. pH. We covered pH in an earlier video. It's the amount of acidic nature of a system or the alkaline nature of a system. We said our blood pH is around 7.3, or in lung tissues maybe 7.38, a little bit more alkaline. The pH in our stomach, because of gastric acid, which is hydrochloric acid, is somewhere around 2. The pH in our small intestine, where all the enzymes that digest our food, that's around pH of 8.4. We have a certain range of pHs that our body is used to functioning at. And we have a bunch of enzymes in our body that function at those pHs. But if you change the pH, if you change the pH of an enzyme, say you had it in a test tube, you were doing an experiment, and you've got a solution in here, and you change the pH, add some acid to it or something like that, you can affect the enzyme ability to function. In our system, of course, we have buffers that prevent that enzyme from, or that system from changing pH, but pH can affect enzyme activity. Again, it can be represented graphically. pH. Let's talk about a system where we've got blood and we're going to have 7.35, which is fairly average for a pH of blood. And that's where, again, rate of enzyme activity up here. We have an optimum and 7.35. That's the best it can get. At a slightly higher pH, it drops off. At a slightly lower pH, it drops off. Temperature, when we had this graph, the temperature graph was not symmetrical. This graph can be. It doesn't have to be, but it can be, because the system can denature on both sides. So you can have denaturing on both sides of this thing. The enzyme just gets too deformed, so it can't work, and just the rate of enzyme activity falls off. Not always true, because it is possible for some enzymes that they will function at a range of pHs. So there may be that a particular enzyme still works at a range of pHs. So this is the case in the case of ripening fruit and things like that, where the pH in fruit changes and the cells are still alive. So there may be a range in pHs like that. But the same sort of thing is true. I'll just go back to the other diagram here. The same sort of thing is true. If you get too far away from optimum pH, the enzyme activity will decrease. Now, I mentioned this business about a test tube on purpose because a lot of this comes from experimental data. How do you measure the rate of enzyme activity? How do you know what's actually happening? In some cases, you might have a gas being given off. And it may be that the system gives off oxygen gas, so you can capture the amount of oxygen gas. So if you change the pH, you can get a certain amount of gas being liberated. Change the pH again, add a little bit more acid to it, it'll alter that, and so on. So you wind up with graphical data. And oftentimes there's data presented, and the determination is, or the question is to determine the optimum pH. So those are two which are conditions that affect enzymatic activity. Don't think I need that. Let's look at the third one. The third one is not really an environmental condition. The third one I want to list is something that could be added to it. I'm not talking about adding a base or an acid or something like that to it. I'm talking about the presence of heavy metal ions. Heavy metal ions, these things, mercury, lead, thallium is a third one. These things are heavy metal ions, big ions. The nucleus of these things are very, very large and very, very positively charged. And you know that you've heard of mercury poisoning and you've heard of lead poisoning. Well, what happens with these things is if you get an ion like mercury in a system, and I'll just use this enzyme again, here's mercury, nucleus, very positively charged. I shouldn't scribble it out, let me draw it properly. Here's an ion mercury, but it's got a huge positive charge because of its big nucleus. I'll just put a bunch of plus signs like that. What that does when it's close to these enzymes is it actually has an affinity for electrons. So this positive charge here is going to draw electrons towards it. So if this actually comes in contact with these or is close to it, the electrons get disrupted. So disrupting the electron positions in these enzymes will affect hydrogen bonding. So the more mercury or the more lead or the more thallium that's in the system, the more this gets disrupted. So people who work in environments where there's a lead level, they get their lead levels tested. And if it's low, that's not a problem. There's always a trace amount of it around. It could be elevated, and that's a problem. So they have to, they take some time away from work for that. That's what lead poisoning is like. Mercury poisoning, lead poisoning is the same sort of thing. The presence of heavy metal ions will affect enzyme activity. The more you get, the worse the situation is. There's more to it than that. We mentioned inhibitors as well. This is the presence of heavy metal ions. Before, when we mentioned the lock and key model, we had the presence of inhibitors. Now, there's two different kinds of inhibitors. I only mentioned one of them already. One of them, the one I mentioned, is the competitive inhibitors. Remember, those ones are the ones that actually compete for the active site. So if I represent this part of the molecule here as the active site, the substrate is normally going to come into contact with the active site and the reaction proceeds from that point. Well, if the inhibitor gets in there instead, then that blocks the substrate. It can't get in, so no reaction occurs. But there's another kind of inhibitor. The other kind of inhibitor is called a non-competitive inhibitor. Sometimes called an allosteric inhibitor. This type of inhibitor literally bonds to the enzyme in a position that's not the active site. You remember the structure of these things is that the outside of these things has got R groups sticking all over it. That doesn't mean that you couldn't have something bonding onto the enzyme in a position that wasn't the active site. So let's take this part here. And something is attracted to the R groups and it actually adheres to the R group. So we could have an allosteric inhibitor here. So why should that have any impact? Well, very similar to the heavy metal ions, the allosteric inhibitor, if you add something here, it's going to cause an electron shift in the whole molecule. So electrons will be drawn towards here. If you draw the electrons towards that, that's going to affect the dipoles in here, that's going to affect the hydrogen bonding, that's going to affect the shape of the molecule, and it may simply cause it to denature so much, the active site will change shape and it won't work. So we've got the presence of inhibitors, two different kinds of them. The fifth and sixth factors, there's 6 of these things that I want to mention, have to do with concentrations. We've got temperature, pH, presence of heavy metal, concentration, I'm going to have to go down a little bit here, of substrate, and the concentration of enzymes themselves. These are almost no-brainers when you think about it. The factors that affect the rate of enzyme activity. If you've got a system, and it's an enzymatic system, and it's supposed to have some reaction occurring in it, the rate of that reaction is going to be somewhat dependent on how much substrate is available. If there isn't any substrate, there's not going to be any reaction. If there aren't any enzymes, there's not going to be any reaction. So you get this kind of a situation here. If we take this as the substrate concentration, and this is the rate of enzymatic reaction here, if there's very little substrate, way down here, there's going to be very little reaction. But as the substrate increases in concentration, you're going to get more and more in reaction, and it'll actually level out. The reason it levels out is because there's only so many enzymes. All the enzymes are busy. So there is a maximum. I mean, cells don't contain unlimited number of enzymes. They contain a certain number of enzymes. They can generate more enzymes, but if all the available enzymes are being used, they're all engaged in activity, then it doesn't matter how much substrate you put in, you're not going to overload it so that it makes it work faster. So there's a maximum level here, maximum rate, which is somewhat dependent on the amount of enzymes that are available. At the same time, if we change this, I put enzyme concentration. If there's no enzymes, there's not going to be any rate of reaction either. And as the enzyme concentration increases, you're going to get a maximum rate. Of course, it's going to level out at some point where there's only so many substrates. If it's so, you know, the enzymes can't react if there aren't any substrates. So these two are somewhat the reverse of each other, to be quite honest. One's dependent on the other. So that sort of takes the information from the previous two videos and puts it all on one place so that you can sort of see how enzymes are working in systems. The next video in this series is more like metabolism, like what's actually going on in a system. We'll take a look at specific metabolic pathways and how that works to keep organisms alive. Thanks very much.